We're now going to pivot away from speciation and just look at the evolution of variation and its importance. So, why is phenotypic variation important for evolution? I'm Mr. W from learn-biology.com, where we believe that interaction and feedback is what leads to deep, substantial learning. We're so sure of that we provide a money-back guarantee that comes with your subscription. To help you study, I've put together a checklist that you can download at apbiosuccess.com/checklist. Phenotypic variation is essential for evolution. It's the raw material upon which natural selection acts. Natural selection selects for organisms with phenotypes that confer a selective advantage. Remember that the genotype is invisible to natural selection. What natural selection sees, what natural selection acts upon, is the phenotype. And the result is that individuals with advantageous phenotypes survive and reproduce at higher rates than organisms with less advantageous phenotypes. Now, as those organisms reproduce, they're passing along their genotype, but that's somewhat of a secondary effect. Natural selection acts upon phenotypes. And note that with no phenotypic variation, there can be no natural selection and no adaptation. And that's why loss of variation is so dangerous to a species and can often lead to extinction. So here are a couple of examples of the importance of phenotypic variation. Let's start with this one. Explain how variations in phospholipid structure can serve an adaptive function in browsing mammals that forage in snowy environments. Phospholipids, we remember those from Unit 1. They've got a glycerol with a hydrophilic head and a hydrophobic tail. These are fatty acids over here. Note that these fatty acids can be saturated or unsaturated, and there can be a lot of variation in that. In mammals that browse in the snow, there is a tendency for the phospholipid tails that are in the body core to be more saturated, and those in the extremities to be more unsaturated. Why? Because as the temperature decreases, these unsaturated fatty acid tails keep the membrane fluid so that diffusion can continue to occur, so that oxygen can continue to flow from the blood into the cells, whereas the phospholipid tails in the body core are more saturated, and that provides a correct level of fluidity in that part of the body. So there tends to be a gradient of more saturated fatty acid tails in the core, more unsaturated fatty acid tails in the extremities. Your success in AP Biology starts here. Are you struggling with AP Bio? With learn-biology.com, students get the skills and confidence to be a top student and earn 4s and 5s on the AP Bio exam, guaranteed. Go to learn-biology.com to find out how you can master your biology course and crush the AP Bio exam. Explain how variation in hemoglobin maximizes oxygen absorption in humans and other placental mammals at various life stages. Let's remember that hemoglobin is the protein that transports oxygen in red blood cells in almost all vertebrates. Before birth, humans and other mammals produce fetal hemoglobin. Fetal hemoglobin is a variant, and it has a higher affinity for oxygen than adult hemoglobin. Here's the difference. All hemoglobins are the same in that they have these two alpha chains. They're shown in red over here, but adult hemoglobin has beta chains, where fetal hemoglobin has gamma chains. They're shown in yellow over here. Because of the higher affinity for oxygen, the fetal hemoglobin will absorb, it'll create a gradient that will cause oxygen to flow from maternal blood in the placenta over here into fetal blood. So here's the gradient, and that of course is highly adaptive because that's how the fetus gets oxygen while it's in the uterus. But after birth, what happens is that developmental genes kick in, and slowly the beta form starts to be produced more and more until it's completely predominant in the adult, whereas the gamma form starts to fall and fall and fall. And you can see that happens even before birth, but the transition is pretty complete 42 weeks after birth has occurred. Explain how variation in chlorophyll types increases the efficiency of photosynthesis. Green plants have two main types of chlorophyll. There's chlorophyll A over here and chlorophyll B over here, and the difference comes down to just a functional group. Chlorophyll A has a methyl group over here, whereas chlorophyll B has a carbonyl group. Chlorophyll B absorbs more blue light, and that's best in indirect light or in shady environments or shady situations. And chlorophyll A absorbs more red light, and it's best in direct light. And therefore, plants can do a couple of things. One is that there are plants that are shade adapted and other plants that are direct light adapted, and those will have different proportions of chlorophyll A and chlorophyll B. And then most plants have both. And having both types of chlorophyll increases the amount of light energy that plants can use during photosynthesis over the course of a day or throughout the seasons. Here are your next moves for AP Bio success. Please subscribe to learn-biology.com and please watch this next video.